Hello, my name is Anna Cavalieri. I'm from the United States, and I would like to introduce myself and share a little bit about my background, experiences, and qualifications. I have nearly 7 years of teaching and tutoring experience combined. My first job was when I was 17. I worked as a university teaching assistant at Florida Gulf Coast University in the subject of Biology 1 with lab. Also at Florida Gulf Coast University, I worked as a college algebra university teaching assistant. Then at the University of Florida, I worked as a molecular genetics university teaching assistant. I've spanned a total of 7 semesters working as a university teaching assistant. Also at the University of Florida, I went on to work as a STEM and humanities tutor for student athletes in the University of Florida Athletic Association. I tutored a multitude of subjects that ranged from psychology, international relations, math, chemistry, biology, and more. When I graduated from the University of Florida in 2023 with a Bachelor of Science in Biochemistry, Bachelor of Arts in French and Francophone Studies, and a minor in anthropology, I then went on to participate in the TAPIF program, or the Teaching Assistant program in France, where I worked as a English language teaching assistant at the secondary school level in a lycée or high school in Colmar, France. This experience was very formative for me as I realized I really enjoy teaching abroad and would like to continue to teach abroad in an international school in the subject of biology. I am also in the process of acquiring my CELTA certificate. As for my personal testimonial and teaching philosophy, I believe that while memorization and structured assignments such as multiple choice tests are important for building a strong foundation in a subject, it is important to give students more challenges to develop critical thinking skills and creativity. This includes facilitating class debates and discussions, having the students give oral presentation on research projects so that they can demonstrate why they know what they know, giving short answer questions on exams, and having free-flowing conversation in class. I also believe that every student is different, and as such, teachers should tailor their teaching style to accommodate the needs of others. This can be done by alternating between more structured lecturing and exams, creative solo or group projects, and working one-on-one with students to discuss their learning progress. I also believe that encouraging group collaboration not only allows students to be more social, but allows them to teach and learn from their peers. While teaching English at a high school in France, I realized that high schoolers have a very small attention span and work best when they are engaged, actively participating, and collaborating with others. However, it is also important to keep them disciplined and introduce structure into their schooling. To answer the question of why I want to work at the International School of Hamburg, I really enjoyed my time teaching abroad at the secondary level for a school year in a high school in France and would love to continue teaching abroad. I also believe that I possess the characteristics for such a job. I am culturally competent. I speak multiple languages in addition to English, such as French, Georgian, and some Spanish. I have also lived in other countries such as France and Georgia. And ultimately, I want to continue to combine my love for biology and science with pedagogy in this role. I am currently a biology research assistant at Florida Gulf Coast University, where I work one-on-one with a professor conducting independent research. I really enjoy this process of investigating real-world problems, and I want to foster this sense of discovery and creativity to students in a classroom. All right, now I will be presenting a teaching sample for a biology classroom curriculum. The question I have decided to work on for this teaching sample is, what is the genetic code? So this is a question I would ask the class. What is the genetic code? Then I will have them answer this question, brainstorm certain possibilities. Some may say DNA, some may say genetic heredity, some may say more specific answers such as DNA nucleotides or nitrogenous bases or even codons, which that's a very advanced answer. So after asking the class to sort of ponder this question, then I would go in with the next question. What is the difference between nucleotides, nitrogenous bases, and codons? Once we have discussed this a little bit, students have given their answers, then I would go on to really break down the differences between these three things and answer the question of what is the genetic code. So, what I would do first, I would draw on the board sort of the broadest idea of what students think of when they're asked this first question, DNA. I would go ahead and draw the double helix structure of DNA as such. This is the double-stranded double helix of DNA, which many students can recognize. And then I would break it down even further. I will say this is the overall structure of DNA, but if we zoom in a little further, what is the structure? Well, if we zoom in a little bit further, we have something called the sugar phosphate backbone and nitrogenous bases. I think it would be helpful to have a reference while drawing this, as it is a little bit more complex. But I am also simplifying it. Instead of drawing the phosphates, I'm just going to represent them as P's. And instead of drawing the bases, I'm going to represent them by their corresponding letters. All right, so we have one side of the backbone complete. We're going to do the other side. On the other side, the sugars are upside down. I will explain this in a little bit more depth after I'm finished. All right, so there we have it. We have our sugar phosphate backbone plus the attached nitrogenous bases. So what does all this mean? We go from this overall structure of DNA into this more zoomed in version. Well, here we have our sugars, and attached to the sugars, we have our phosphate groups. This is what we mean by sugar phosphate backbone. And then because DNA is double-stranded, so that it makes this double helix shape, we have two sides, two sugar phosphate backbones. Then attached to each unit of the sugar phosphate backbone, which is one sugar molecule attached to one phosphate group, we have a nitrogenous base. I've represented the nitrogenous bases by their corresponding letters. There are 4 of them: adenine, thymine, cytosine, and guanine. Now, I will take a moment to ask the class to consider this question again. What is the difference between nucleotides, nitrogenous bases, and codons? Well, if it's not already clear, I will reiterate that this unit right here, the sugar plus phosphate group plus a base, is a nucleotide. So this unit right here, phosphate group plus sugar plus base, is a nucleotide. The nitrogenous base itself is either adenine, thymine, cytosine, or guanine. These, the nitrogenous bases, are the most important aspect of this whole scenario, as they are what carries the information. It is what's passed down from parent to offspring in a special sequence called the genetic code. So in other words, the nitrogenous bases are the fundamental unit of the genetic code. They consist of the four chemical bases: adenine, thymine, cytosine, and guanine. If we're talking about RNA, then the thymine will be replaced by uracil. So in RNA, thymine becomes uracil instead. So I'll add that here as a little note. Thymine is instead uracil in RNA. These are the bases for DNA, and while DNA is in some ways the most important part of the genetic code, as it is a structure that holds, the original sequence, the original bases. This is an important linkage because DNA is then transcribed into RNA, and then the RNA is then translated into protein. So it is important to remember RNA has uracil instead of thymine. All right, and then once we've established all of our bases, then we can assemble our bases into something called a codon. A codon, right here, that term right there that we introduced, a codon is an assembly of three bases. For example, we have AUG, adenine, uracil, and guanine. Bonus question, is this RNA or DNA? Students would then say this is RNA as we have the uracil instead of thymine. This is what we call the start codon. This is the start codon of RNA, the RNA transcript, and it codes for the amino acid methionine. Methionine, which we can represent as an M for short, or just an M, or just MET would be another way, MET. But let's just write it out here so you can see methionine. This is an amino acid. An amino acid is a building block of a protein. And proteins are important as they build cells, they build entire tissues, they build entire organs, they have a multitude of different functions. So building proteins is the ultimate goal of the genetic code. The genetic code carries sequences, and then those sequences are used to build things for the cell, for the body. Now, AUG is not the only codon there's other codons. The only important thing to remember is that three bases confer to one amino acid. So the ratio here is 3 to 1. 3 to 1 is an extremely important ratio to remember. 3 bases for every one amino acid. Now, if we revisit this question, nucleotides versus nitrogenous bases versus codons, what makes up the genetic code? I will go back and ask the class this. What makes up the genetic code? Some may say nitrogenous bases, and this would be, I think, the most technically correct answer, as nitrogenous bases are the most fundamental unit of the genetic code, these 4 bases right here, as well as uracil. But these 4 bases right here, these 4 original bases, are the fundamental unit of the genetic code. And then when you assemble them into a codon of 3 bases, every 3 bases codes for 1 amino acid. And that would be a more complex answer. Students may also argue that they believe nucleotides are what make up the genetic code, as nucleotides also include the entire sugar phosphate backbone, and the sugar phosphate backbone protects the bases, which I could also consider a correct answer as long as they explain why they believe nucleotides are part of the genetic code. So there really is no right or wrong answer. It's really the level of complexity that they want to, how far zoomed in they want to go. Do they want to go the level of DNA and its basic structure of, sugar phosphate backbone attached to bases? Do they want to look at the level of bases themselves? Do they want to even assemble them into codons? It's really up to the student to decide, as long as they can back their answer up, give evidence for why they believe what they believe, and how they can explain how their answer fits into their understanding of the genetic code. When I'll be grading students for this answer, I'll mainly be looking at 2 things. I'll be looking at how well can they back their answer up, how well can they defend why they believe a certain term or a certain unit makes up the genetic code. I will analyze how well they understand the subject material, how well they understand the sort of flow from a larger sort of zoomed out version of the genetic code to a more zoomed in version of the genetic code. And I will also be looking at creativity. I'll be looking at their unique take on the genetic code, their unique take on what they consider to be the core aspect of the genetic code. I'll also be looking at general interest, general mastery of the subject material, in general, if their answer is complex and delivered well. That wraps up my teaching sample for teaching a class on the genetic code. Thank you for listening.